Today we are discussing about giant chromosomes. Giant chromosomes are special types of chromosomes. In some organisms, there are special tissues in which chromosomes undergo structural specializations. Such specialized chromosomes include the giant chromosomes and the B chromosomes. Today we are discussing about the giant chromosomes. Giant chromosomes are the exceptionally enlarged and highly specialized chromosomes found during some stages in the life cycle of certain cells. Two kinds of giant chromosomes have been recognized: polythene chromosomes and Lamb-Rush chromosomes. Polythene chromosomes are the cable-like multi-stranded giant chromosomes formed of a bundle of many identical chromatids. They are formally called as salivary gland chromosomes because they are first observed by Balbiani in the salivary gland cells of the larva of Chironomus. Chironomus is a fly whose larvae spend their life in ponds and slow streams. Polythene chromosomes are found in the synergids and antipodal cells of the ovules of certain angiosperms, in the macronucleus of some ciliate protozoans, and in the salivary glands, intestinal epithelium, Malpighian tubules, fat body cells, ovarian nerve cells of some diphtheria larvae like Drosophila, Chironomus, etc. This is the picture of a polythene chromosomes. These are chromosomal puff and dark bands and inner bands. The number of polythene chromosomes in diphtheria flies is always haploid in relation to the normal somatic number, but in ciliates their number ranges between 25 and 50. In diphtherians, homologous polythene chromosomes occurs in somatic pairs. That means they are in permanent prophasic conditions. In between the companions of each pair is a longitudinal slit known as pairing gap. In ciliates, polythene chromosomes do not exist in somatic pairs but remain distinctly separate from one another. Next, we are discuss about the diphtherin polythene chromosomes. Diphtherin polythene chromosome consists of many extended chromatids produced at the repeated replication of a single chromatid without accompanying nuclear duplication and cell division. The chromosome has a definite banding pattern of organization. It has a characteristic banded appearance with a series of dense and dark bands alternating with light interbands. Nearly 85% of the chromosomal DNA is in the dark bands and the remaining 15% is in the inner bands. Bands are mostly heterochromatic. Heterochromatic means the DNA is highly condensed, and the inner bands are euchromatic. Euchromatic means they are very less condensed, but they are transcriptionally active. It is assumed that the band region is formed by the merging of the chromosomes of adjoining chromatids through the condensation and localized supercoiling of the chromatin filaments. In most cases, the bands completely encircle the chromosomes, forming a circular disc. The bands are polygenic and each of them is believed to contain many tissue-specific genes. Each gene may have multiple copies which are not transcriptionally active all the times. On the other hand, interbands are believed to contain housekeeping genes which are constantly transcribed and translated into the products necessary for the cells. Next are puffs. At some stages in the larval growth of insects, one or more specific bands enormously enlarge and bulge out, forming local swellings called chromosome puffs. They are formed by the uncoiling of chromatin filaments. Puffs are the transcriptionally active expansions or extensions of chromatin. So they are active centers of synthesis and accumulation of RNAs. In some cases, in addition to the RNA, extra DNA is also produced during puffing. Such puffs are the sites of specific gene amplification and are responsible for differentiation and development. In some puffs, chromatin filaments are drawn out as a series of lateral loops known as Balbiani rings. These are Balbiani rings. It shows the inner bands and they are dark bands. There are four categories of chromosome puffs as follows. First one is stage specific puffs. Second one is tissue specific puffs. Third is constitutive puffs, and fourth is environmentally induced puffs. Stage-specific puffs are which appear during a particular stage of development, such as molting. Tissue-specific puffs are active in one tissue but not in others. The constitutive puffs are active almost always in a specific tissue. Environmentally induced puffs means that they are appear in response to an environmental change, such as heat shock. Puffing is a cyclic and reversible process. At definite times, puffs appear, grow, and finally disappear to repeat the whole cycle over and again. Puffing involves the regular succession of four major events, namely excessive accumulation of acidic proteins. Second is the detachment of DNA found basic proteins to expose the DNA for transcription. The third step is despiralization and decondensation of chromatin. Fourth step is the active synthesis and storage of RNAs. It is believed that puffing is initially induced by the accumulation of acidic proteins at specific regions. It has also been shown that the insect molting hormone ecdysone induces puffing. Puffs and Barbiani rings are regarded as the active centers of DNA duplication and RNA synthesis. Hence, puffing is interpreted as a manifestation of gene expression. Since active DNA duplication occurs in puffs, they may contain heavy accumulations of redundant and repetitive DNA sequences. Next is endomitosis. The enormous size and polytony or multi-strandedness of diphtheria polythene chromosomes are the result of a peculiar kind of chromosomal reduplication called endomitosis, or it's also known as the endoduplication. Endomitosis is the repeated duplication of chromatids in geometrical progression without accompanying the cell division. In this process, the DNA and chromatids undergo many rounds of replication without spindle formation, disappearance of the nuclear envelope, separation of daughter DNAs and daughter chromatids, and the division of the parent cells. Consequently, the quantity of the DNA and the number of chromatids keep on multiplying in geometrical progression. Thus, endomitosis makes the chromosome multi-stranded and increases its DNA content nearly 1000 times or more. Endomitosis is quite common in certain types of cells of plants and animals. Examples are human liver cells, giant abdominal neurons of aplasia, and the macronucleus of ciliates. Next is the polythene chromosomes of ciliates. As we discuss about the polythene chromosomes, the diphtherians, we are now discuss on the polythene chromosomes of ciliates. In ciliates, polythene chromosomes neither contain puffs and Barbiani rings, nor do they show any signs of transcriptional activity also. In them, fully formed chromosomes first get transected by membranous partitions which appear in interband regions. These membranes consist of only a single diffuse layer of proteins. The formation accompanies the polythenization of chromosomes, then each band and the adjacent interband get completely engrossed within a membrane, forming a vesicle. Thus, a series of vesicles are formed all along the chromosomes. The vesicle stage would be followed by the shrinkage of the chromosomes. The transaction of the chromosomes, the formation of vesicles, and the shrinkage of the chromosomes are associated with a progressive reduction in DNA content. Nearly 80 to 90% of the DNA is degraded to acid soluble products that are permanently lost from the chromosomes. Soon after the degradation and elimination of DNA, the vesicles disappear. The small amount of DNA that still remain undergoes repeated replication. Next is Lambrusche chromosomes. Lambrusche chromosomes are the highly extended type of gen chromosomes with a series of paired lateral loops all along the length of a central axis. The whole structure looks like a Victorian Lambrusche. That's why it is known as the Lambrusche chromosomes. Lambrusche chromosomes are the longest of all chromosomes. Their enormous size is attained by the growth of chromatids. Lambrusche chromosomes were first observed by Fleming. They are characteristically found in the diplotin stage of the primary oocytes of insects, sharks, amphibians, reptiles, and birds. They may be longer than polythene chromosomes. The Lambrusche chromosomes found in the oocytes of the salamanders are the largest of all known chromosomes. A Lamprecht chromosome consists of a central axis and paired series of loop-like lateral extensions. The central axis consists of two bivalent homologous chromosomes, each with two chromatids. So, altogether there are four chromatids. They are held together through contact points or chiasmata. The chromatids are drawn out into paired lateral lobes. So, the central axis extends to the lateral lobes as loop axis. Loop axis is only a single chromatid of one of the chromosomes. Lateral lobes contain bundles of submicroscopic fibers also. Both the central axis and loop axis are formed of deoxyribonuclear proteins. The loop axis may be coated with a matrix of RNA and proteins. Lateral loops contain a fully extended DNA duplex. They are believed to be formed by the uncoiling of the sister chromatids, making their DNA available for transcription. Lateral loops are active centers of RNA synthesis. Each loop may contain one or more transcription units. Most of the newly synthesized RNA molecule associate with proteins and form in ribonuclear protein matrix. But some of the mRNA molecules play an active role in the subsequent development of the zygote. The chromosomal axis may contain paired series of granular chromosomes. It is from this regions that the lobe axis extends out. Usually 1 to 9 lobes may arise from each chromosome. Small swellings without lobes may be present toward the end of the central axis. They represent the telomeres. Each bivalent may contain a lobe-less swelling also. It represents the centromere. According to the master and slave hypothesis of Callan and Lloyd, each loop axis is a polygenic with repetitive DNA sequences and multiple copies of the genes. The centromere is believed to contain the master copy of each gene and the loop axis contains its duplicate copies. Only the master takes part in the transcriptional activity. Lamprecht chromosomes are believed to govern the synthesis activities of growing oocytes. It is suggested that they are actively involved in the synthesis of RNA, proteins, and the deutoplasmic reserves. This is the Lamprecht chromosomes. It represents the chiasma. This is the magnified region of the loop.